Right. Hello?
Water potential, osmotic potential,
Right. What?
Osmotic potential, which are some terms like analysis negative and more negative. Water potential.
Yes, you can use them.
Okay, thank you.
But anyway, before we proceed to talk about transpiration, I just want to give you a summary of what we talked yesterday. What we talked about yesterday. Otherwise, we may get lost. Previously, we have been talking about the importance of water in plants, under plant and water relations. Basically, some of them are, you want to transport solutes within the plant. That transportation of solutes, which are used by the plant, is via the water medium. The other thing is that there are biochemical reactions which can take place within the plant, which facilitate the growth of the plant. Medium to facilitate these biochemical reactions. Now, because a living system is supporting a lot of biochemical reactions, sometimes you get a heat being released in plants to the extent that this heat may start interfering with certain processes. So there's a need to cool the plant using water. So these are some of the in a living system or in a plant. The other issues which we talked about were focused on water itself. What is water composed of? We said that water is composed of oxygen and hydrogen. These elements are joined together by covalent bonds within the water molecule. We have got hydrogen being joined to oxygen via the covalent bond. But if you look at the water molecules themselves, they are also attracted to other water molecules via what are known as the hydrogen bonds. So the hydrogen bonds unite water molecules so that they form this liquid which we know as. Key issues to remember is that you need to understand what is a covalent bond and its importance. We know it facilitates the union between oxygen and hydrogen within the molecule of water. And hydrogen bonding facilitates the union of water molecules so that they create this liquid which we know as water. Then we also talked about the movement of water molecules. Number one, we explained what is known as diffusion. And we said that diffusion is the movement of molecules from a region of high concentration to a region of low concentration. And I demonstrated using a certain diagram showing a passage which can facilitate diffusion. So diffusion, movement of water molecules or molecules of a substance from a region of high concentration to a region of low concentration. Then we also talked about osmosis. We said that in osmosis, you need the two liquids to be separated by a semi-permeable membrane. On one hand, you have a strong solution. On the other hand, you have a weak solution or pure water being divided by this membrane. Then what is going to happen when you have such a situation? You have water molecules moving from pure water, crossing the semi-permeable membrane into the concentrated solution, causing dilution. And that process is known as osmosis. In other words, water is moving from high concentration to lower concentration. The lower concentration of water is in the solution. The high concentration of water molecules is in the pure water or dilute water. But of course, what differentiates osmosis and the diffusion is that with osmosis, you need a semi-permeable membrane to separate the two liquids. And with the diffusion, you don't need any impediment to facilitate the movement of these molecules. Why should the molecules move? This depends on the energy they carry in the form of heat. So if we change temperature, we can actually increase the motion of these molecules and facilitate these two processes. Then we said that in order to understand the movement of water molecules, there is a concept which we call water potential. This water potential can enable us to understand why molecules move from one region to the other region. And we said that a substance or water is what is known as water potential. And this water potential is represented by a symbol known as psi, which looks like a fork. And that psi is a sum of three components. The first component is what is known as the solute potential. Now, the solute potential is a result of actually putting something in water to make a solution, like sodium chloride. It could be sugar. When you are making that solution, you create what is known as the solute potential. Before I explain the other components, I should say that the water potential of pure water is 0 megapascals. That is the measurement of the units. It's MPa megapascals. So it is 0 when it is pure water. When you dissolve something in water, you create solute potential, which is below 0 MPa, it becomes negative. The other component is pressure potential. This is the force which is exerted inside the cell, which can push the cytoplasm against the cell wall. So that is the pressure potential with respect to plant cells. Then we also have another component which is known as the matric component. This is when you have water being attracted to the to the other particles. So that is the matric potential. So water potential has three components: solute potential, which is represented by psi pi plus pressure potential, which is represented by psi p plus matric potential, which is represented by psi m. So the water potential will explain how water moves in the plants. Later on, when I talk about the transpiration, I will be using some of these terms. The last thing which we talked about is the cell condition. The plant cell condition when it is surrounded by solutions. The first condition is when you surround the plant cell with a very strong solution. This can make the plant cell lose water, of course, through osmosis, and the cell will go into a state of plasmodysis. That is, the cytoplasm inside the cell will shrink as a result. And we call this condition plasmodysis or hypotonic condition. You are surrounding the plant cell with a strong solution. If the concentration inside the cell is equal to the concentration outside the cell, you have water going inside the cell and also water going outside the cell, almost at equal rates, and it creates a flaccid condition. This is what we call the isotonic condition, because what is inside the cell and outside the cell is equal in terms of concentration. The last thing which we talked about was the hypotonic condition, where you have a dilute or a weak solution surrounding the cells, and you have got a movement from the solution into the cell to create this hypotonic condition, and it creates turgidity, which is important for the structure of the plants. So this is what we talked about yesterday. That is, in a nutshell, that is the summary. So we are now going to proceed.
Noah, can you mute your
Yeah, somebody should mute their mic. Okay, so that is the summary. I don't know whether you have any questions about what we talked about so far. I have highlighted the key issues. Any explanation which you may want on the previous things which we talked about? If not, I'm now going to present on the transpiration, because basically you will need some of the knowledge which we acquired in the previous lectures. So I will start to present on a transpiration. So let us focus on the transpiration process. What is it? Basically, transpiration process is the movement of water from the soil via the roots, stem, leaves, and finally into the atmosphere. So that complete process is the transpiration. Here on this picture, I've got a tank. If we are going to put some water inside this tank, we are going to pump water from the soil, presumably from a bowl, and lift the water into this tank. In the background, you can see there are also trees, which could also be doing the same thing. But the trees will be pumping water into the atmosphere. Their water is not collected in a tank, but the principle is the same. Please note, I am not saying that if you pump water from a bore and put it into a tank, it becomes a transpiration. No. I'm just going to explain that the transpiration process is more or less similar to what we do when we pump water from our bores into the tanks. Here again, I've got a background where we've got this big tree, which is pumping water from the soil into the atmosphere. And in the background, there's also a tank there where you have somebody who is actually pumping water from the soil, storing it into the tank. Basically, what the tree is doing and what that pump is doing are more or less the same. They are lifting water from the soil against gravity up to a certain point. So basically, we've got this plant, which has got roots and leaves. What I want you to appreciate is that in the soil, we have got high water potential compared to what is in the atmosphere or in the leaves. We have got low water potential in the leaves and in the atmosphere. So the direction of water movement is from the soil up into the plant and then out of the plant into the atmosphere. What is important at the base is the root conductance to liquid water flow. Then in the leaves, we have got what is known as stomatal conductance to water vapor flow. Then outside the leaves, we have got the boundary layer conductance to water vapor flow. At the boundary layer, the water vapor is now in the atmosphere. It has escaped the plant. and it has to move from a region of high concentration to a region of low concentration. Here again, I've got the structure of the tree. What I just want to indicate is that at the roots, we have got high water potential. And in the canopy and the leaves, we have got low water potential. So the direction of water flow is the blue route. So water moves from the soil up the tree, and then it is lost into the atmosphere. At this point, I just want you to get that concept. Later on, I will be talking of some of the details as we move. In fact, this slide summarizes what basically we are going to talk about. So when we have finished this topic, if you want a summary of what we have talked about under transpiration, you will get it on this slide, everything. So to appreciate the definition of transpiration, I will repeat again using this diagram. It is the movement of water from the soil via the roots through the stem and out of the plant via the stomata of the leaves. So the whole process, starting from the roots up in the stem and in the leaves and then out into the atmosphere, is what is referred to as a transpiration. So let us focus on the movement of water into the plant roots. Why should water go into the plant roots? Now, the surrounding water, the water which is surrounding the roots, is what is known as high water potential compared to what is inside the roots. Therefore, water will then move from high water potential, which is outside in the soil, into the roots where we have a low water potential. This is the reason why the water flows inside the roots. Remember, the root is concentrated solution. So the water moves into the root. Now, when the water continues to flow into the root, it creates what is known as root pressure or hydrostatic pressure. That is what is created by the movement of water into the roots. Now, I want to differentiate transpiration and what is known as mutation. It's another process where plants can actually lose water through the mutation process. Water can ooze out of special opening called diadathodes. These are found on the edges of the leaves. And this process only takes place during the night. Loss of water via the diadathodes. The one which is referred to as mutation. So if you observe a plant leaf at the edges, you may find some droplets. And this happens during the night. You may think it's dew, but this is not dew because this is coming from the plant. So this is loss of water via guttation. So back to transpiration, we want to talk about the movement of water across the root and the xylem vessels, which are found in the stem. Water moves down the gradient across the cells into the xylem vessels. So there should be high water potential somewhere and low water potential somewhere to create this movement. So there are two distinct pathways which are followed by water as it travels through the root from the soil. Number one, we have what is called the symplastic pathway. In the symplastic pathway, water travels across the living cells, as opposed to the apoplastic pathway, where water moves through the non-living system. And this is the common route. I will have to use a diagram to illustrate the two pathways so that you can understand. Okay, first I'm displaying a situation in the soil. This is what we call the root hair, which is part of the root, which is in contact with the water. So the root hairs are directly in contact with the water in the soil. So because of differences in water potential, water can go into the root hairs, then into the root. Now about the two pathways which can be used. Let's suppose water has entered the root. If it travels across cells, going through the cells, it will have followed what is known as the symplastic pathway, because it is traveling across the cells, going via the cells themselves. But if water avoids the cells and goes via the cell walls, avoiding going through the cells, that pathway is following a non-living system, the cell walls. It becomes the apoplastic pathway. So the plant can acquire its water via two systems, the symplastic pathway as well as the apoplastic pathway. Now suppose the water has traveled through the root and it has reached the stem. We want to understand how the water is lifted from the bottom of the stem up to the leaves. What pushes that water? Remember, there is gravity which can pull the water downwards. Somebody yesterday highlighted the gravity component when we were talking about water potential, and I said, let us ignore it for the moment. But gravity can pull down water, and this water has to go up against gravity. So in order to understand why water has to go up the stem, we have to use the water potential concept. So water movement is a result of water potential gradient between the root and the air boundary layer surrounding the leaves of the canopy of the trees or plants. In fact, there is a soil-water continuum, which is the continuous system made up of water columns from the roots up to the leaves. The other things which we need to understand if we are going to explain why water moves from the soil to the leaves, there are cohesive and adhesive forces which literally move water up the xylem vessels. There's what is known as the cohesion tension theory of sap ascent, which can help us to explain why water should go up in the stem. Now, water evaporation in the leaf generates negative pressure. So at the leaves, we have evaporation of water taking place. And this can create, or this can reduce water potential. So let us focus on adhesion. Remember, we wanted to find out how water can move upwards through the stem. Adhesion is the force between water molecules and the cell walls. And this can actually generate capillary action or movement of water upwards through thin tubes called the xylem vessels. So capillary action is generated by adhesive forces between water and the wall of the tubes. However, I should say that adhesion can force water upwards, but it cannot exceed 1 meter. Although it can contribute, but it cannot exceed the 1 meter. So it is not sufficient to explain the movement of water up to the leaves for large plants. But for short plants, probably adhesion is sufficient to explain what is happening. But the cohesion tension theory can actually account for the actual movement within the stem up to the leaves. If we want to understand the movement of water, probably let us go back to the water itself. What is it made of? Water molecules attract each other via the hydrogen and the oxygen atoms. In fact, we call these cohesive forces because we have got hydrogen bonds between oxygen atoms. If you take one molecule of water and another molecule of water, they are attracted via the hydrogen and oxygen atoms. We call this hydrogen bonds. But a lot of these can become cohesion. And this causes tensile strength. Osmosis, which we tried to explain earlier on, plus evaporation at the top of the trees can create negative water potential. Now, osmosis will be the movement of water from one cell to the other via the membranes. And when the water reaches the leaf surface, it will eventually evaporate. And this creates the negative water potential. This can actually create a pool which extends down the tree trunk or the stem and the roots. And this actually facilitates the loss of water from the plant. There's also another factor which is known as the water meniscus. I know we can demonstrate it in a measuring cylinder as a curved surface when we pour water in a measuring cylinder. Probably you need to know that the water meniscus also is present in the leaves and causes tension, which can actually pull the water through the stem. So the water column in the stem is under tension because of the water meniscus, which creates tension and moves the column upwards. Now, sometimes we may have blockages of water movement where we have xylem vessels being blocked by air bubbles. We call this embolism. It's like airlock. So this is the water in the cells of the leaves, which can create surface tension, which can also add forces to pull water via the stems. Just imagine the leaves, and you have a lot of these forces. If you integrate these forces, it becomes a big force, which will actually pull the water via the stem. Now let us talk of water which has reached the leaf. What is actually going to happen to it? Water can form a thin layer around the mesophyll cells. Water can evaporate into the air spaces. You should remember that water exits the plant via leaves by osmosis. Then via the stomata, due to low water potential in the air surrounding the leaf. Now, as long as the water is moving within the cells, it is the osmosis which is operating. But as the water leaves the stomata, there's diffusion. This will pull water up the xylem vessels. This continuous evaporation of water will cause low water potential, and it creates a gradient which forces water upwards. It will actually be like a pump, which is pumping water upwards. Here I want to focus on the, okay, this is the leaf and this is the stomata. Water within the cells may travel via osmosis as the water escape into the air spaces. It leaves the leaf by diffusion into the atmosphere. Now I want to focus on what is happening here at the stomata. I'm going to explain the opening and closing of the stomata because it can regulate what enters the plant or what leaves the plant. So what regulates the opening and closing of the stomata is the light, number one. Number two, it is the internal water status of the mesophyll cells, because it can give us an idea about the magnitude of water potential, because we know water potential can create a movement of water from one place to the other, depending on the magnitude of these water potentials. We'll also have the abscisic acid, which can also regulate the opening and the closing of the stomata. Now, during the day, I want to talk what events can happen to the leaf, or what the stomata can do. Now, during the day, we have got light, which can influence the accumulation of potassium in the gut cells. And we've got an enzyme known as ATPase on the membrane, which can pump hydrogen ions out of the cell. Now, the process of moving potassium and hydrogen will require energy in the form of ATP, which has to be converted to ADP plus phosphorus, Pi, in order to move potassium and hydrogen in opposite directions. So what is happening during the day is that potassium is pumped into the gut cells. And on the other hand, hydrogen ions are pumped out of these gut cells. The gut cells constitute the stomata. Now, inside the gut cells, we've got malate and chlorine ions. These will combine with the potassium, which will have entered the gut cells, to form salts. Now, these salts will lower the water potential. In fact, it becomes solute potential, which is lower than the water potential of pure water or dilute water. So the entry of potassium into the gut cells, combining with malate and chlorine, will form salts which will lower the water potential. This means that once the water potential has been decreased in the gut cells, water will then move from the surrounding mesophyll cells into the gut cells until they become turgid. Once these cells become turgid, they open. This can now explain why during the day the stomata becomes open. This is the mechanism of opening the stomata. And then what happens during the night? This enzyme ATPase will pump out potassium out of the gut cells into the surrounding mesophyll cells. Actually, the opposite is now happening. Hydrogen now is pumped into the gut cells. The water gut cells is higher than what is in the surrounding cells. So water will move out of the gut cells. And the gut cells become flaccid. Thus, they will close the stomata. So this is how the stomata is closed during the night. Later on, when we talk about photosynthesis, it may help us to understand what is actually happening. But for the time being, this is how the stomata is regulated so that it can actually allow water to escape or to not to escape. Now let us look at the role of abscisic acid. Now, this can help the plant to survive drought conditions. Because when it is dry, the stomata has to close to prevent water loss. So what happens? Abscisic acid will combine with ATPase and pump the hydrogen into the gut cells. Because normally the stomata is open during the day, but the opposite is now going to happen because of abscisic acid. It's going to pump the hydrogen into the gut cells as if it is now during the night. It's a mechanism of controlling opening and closing of the stomata. So when the hydrogen ions go into the gut cells and the potassium is removed, you are actually decreasing, you are actually increasing the water potential in the gut cells compared to the surrounding cells. So water will move out of these gut cells and become flaccid. Once the gut cells are flaccid, the stomata will close. After the closure of the stomata, the water will not escape the plant. In other words, we are now breaking the transpiration process. We are more or less stopping it, because if that transpiration process is allowed, it will kill the plants, because the plants will lose a lot of water. So under drought conditions, the plant will respond via abscisic acid so that it can stop this transpiration, because it's going to damage the plants. So plants are quite sensitive to environmental conditions. So this is the open stomata. You can see water is going in, potassium is going in, and you have an opening here. When the stomata is closed, everything is leaving the gut cells. Water is getting out, potassium is getting out, but we know hydrogen is going in the gut cells. So this is how the plant can control what goes inside it and what leaves it. Right. We can use what are known as porometers to understand the water status in a plant. We can measure stomatal conductance using this instrument. This is some of the old porometers. You know, it's basically a computer with a peg. You know, this is where you clip the leaf. Then this machine is going to tell you a lot of things about this plant related to its water status. It will tell you whether the stomata is open or it is closed, how much water is being lost, leaving the plant. You can measure it. Nowadays, we have got, you know, portable porometers. You know, this is a big machine, quite heavy. But because of, you know, advancement in science, we now have, you know, smaller. And, you know, these are easy to use. Porometers. This looks like a peg, a clip. You just clip on the leaf. Then this, you can read off water which is being lost at that moment. It can tell you whether the stomata is closed or open. If it is closed, probably it has something to do with the moisture environment. If it is open, probably the plant is in a healthy state and everything is okay.
Sir,
Lastly,
sir,
yes,
someone is presenting at Yazukona slides.
is
Kelvin Musa.
can you see this?
No, Kelvin Musa is presenting, so I share one in.
Or somebody is presenting.
Yes,
What is he presenting?
nothing.
How can we stop him?
Kelvin Musa, please stop presenting.
Stop sharing your screen, Musa.
Ingomburitsa ise tiendere.
Okay. Are you seeing the slides now?
No,
No.
hatisi kutomboona sekungoona Kelvin.
Anyway, let me finish. Because I recorded this, but you are hearing what I am saying.
Yes.
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 Yeah. Now I am talking about factors affecting transpiration. First, we've got temperature. Increasing temperature can increase evaporation of water on the leaf surface. Now, it decreases the water potential. It also increases the rate of diffusion. Water molecules have more kinetic energy. At high temperatures, ABA may close the stomata and reduce transpiration. Factor number 2, air movement or wind. Faster air movement removes the water vapor and decreases the water potential. This increases transpiration. Still, air can decrease transpiration. The other factor is humidity. That is the amount of water vapor in the air. Now, high humidity will slow the transpiration process, but low humidity can increase transpiration. So this can decrease the water potential above the leaf surface. Third is light intensity. Light increases the rate of photosynthesis. It can increase amount of glucose in the leaf guard cells, leading to low water potential. This causes water to enter guard cells and cause the stomata to open. Now, this can increase water loss via the stomata. So there are 3 factors: humidity, light intensity, and the temperature, which can affect transpiration rate. Now, you may find that plants have got to be adapted in order to control some of the effects of transpiration. Some plants have got sunken stomata, so that they create high humidity to sort of slow down the transpiration rate and loss of water. Some plants have got thickened and succulent leaves. Others have got the presence of the crassulacean acid metabolism or the CAM system. Using this system, stomata can only open at night when they fix carbon dioxide as mallet, and the stomata may remain closed during the day to avoid evapotranspiration. I will talk about this later when we discuss photosynthesis. Well, these are some of the plants which are adapted to deserts where transpiration can actually kill plants. So these are some of the cactus plants. Now, measuring water potential, we can use tensiometers, which are put in the soils. We can use pressure chambers directly on the plants. So this one is a tensiometer. This is the pressure chamber where gas is forced into a chamber. Then we take a reading. Now, water potential can be measured, and it can tell us the plant status or the physiological condition of the plant. For example, dehydration can cause a lot of changes within the plant. So if you measure water potential, it can tell you about what is happening with the photosynthesis, stomatal conductance, protein synthesis, etc. So water potential is very important measurement which can tell us about the condition of the plant or the healthy status of the plant, if you want. So this is it. This is the end of the lecture on transpiration. I will stop sharing. Okay. We have finished the lecture on transpiration. Any questions? Hello, yes, Tungesai Kadzilange. Kadzilange, you can go ahead. Hello? Tungesai, are you there? You want to say something? I can see your hand up.
Yes.
Yeah.
Water potential with reference correction in Basere.
Kuti?
With reference correction in Basere. Water potential. Water potential with reference correction in Basere by equation.
Well, it's not really important at this stage.
Okay.
We're in the area, I got.
I want this. Thank you.
Any other issues? Anyway, for your information on the water potential, I just want you to understand how we can use this concept to explain the movement of water, especially up the stem to the leaves.
Sir,
Yes.
there is a point that say the decrease in solute potential increases the water uptake. I think I am a bit lost there. How does the solute
Soil
potential
or water potential?
solute potential? The point says solute potential.
Yes.
It says a decrease in solute potential increases the water uptake. I'm confused there. How come the solute potential decreases, then the water uptake increases?
Right. What you should understand is that if you take pure water, right?
Yes.
It has got 0 MPa as the water potential.
Yes.
Now, if we add the salt to that pure water and create a solution, we are going to get a negative MPa.
Okay, sir.
So we are going to decrease that water potential. Now, if we separate the liquid where we have added water by a membrane, on the other hand, we have the pure water separated by a membrane, and on the other side, we have got a solution. We can create water movement from pure water going into the solution via that membrane.
Okay, sir.
Yeah. Basically, that is what is happening.
Okay.
So
Thank
inside
you.
the root, we have got some salts there, and there's a negative water potential. Outside the root, of course, we have got a weak solution of water in the soil with the high water potential. So there's a flow from the soil into the root.
Okay, thank you, sir.
Another person? So tomorrow, what time can we meet?
It's 9.
Oh, there's no mathematics at 9.
And they're going to do it, they've done today.
Say, I think 15 hours or.
So meet at what time?
It's 9.
At 9, okay.
No, I'm waiting it in the mates, guys.
Mates munopedza nenguva.
Hatisitomboziva kozi vamwe vari kutoita izvozvo hatisitomboziva watanga na9.
Hello.
Yeah, let's try to adjust your more possible benefit.
Yeah, go ahead.
To do PPST. So for tomorrow, just
Can you write? Is it tablet? Yeah, tablet, can you write?
in.
Then after tablet, will get to you. Okay, you can come in.
Okay, I was saying PP is supposed to do 11. So I was thinking if it is possible, starting tomorrow we stick to the timetable so that mathematics can also benefit from the lecture.
Yeah, so I'm asking you, what time do you want me to teach you?
At 11, say.
Yes.
At 11.
11 is
Because,
okay.
yes. As for me, I can teach any time, you know, whether midnight or whatever time which you want. I'm available. What is important is for you to tell me when I should come and teach.
Guys, paneling lesson genetics na 11. Auntie, guys.
We're supposed to do PPST.
Genetics.
So if you are doing so you can come in at 2.
Corner 8.
Yeah.
Guys,
nobody kana 9 guys tobva tapinda mumesi pane mesi sepa mwana ngwaza. Say I think
Okay, so we can do it at 2.
Kana 2.
Yes.
Better.
Let us start at 2. 3 is a bit late.
Yes.
So tomorrow we need at 2.
That's okay then.
yes, sir.
Yeah.
Agreed. Yeah, good.
Yes.
So that we don't interfere with other processes. We will need your mathematics, you know, to sort some of the issues here. Okay, another question? So after this transpiration, I think I will give you a test on what we have been talking about.
Test
Very
when?
Yeah, sometime pakati papa I will post it. I will see how I'm going to administer it.
Nyarara itu zvakakosha.
Before you write your examinations, are you going to have a revision week when you are on campus?
No.
Guys, can you mute your give me your mic?
Say,
Yes.
Patishokuro kana live isingaitika munobatisawo kuti haaveringa apa apapera futi.
Right. If you were listening to what I was saying, that's where your test comes from.
Oh,
Tanga, I summarized what we did.
yes.
So each time I come up and do a summary, and that is where your questions will be. For example, will you be able to explain what is known as adhesion? Do you know what is adhesion?
Yes.
Do you know what is cohesion?
Yes.
If you know those things, then those are the things which you ask you. Hypotonic condition. Do you know what is hypotonic condition? Capillarity. So those are the things which we ask.
Tafadzwa makoni nyarara.
So I will ask very direct questions. Yeah. Saka, anyway, I think we are now through, but you can always, yes.
Mune zvavakakotipawo here ndasabatikawo
Hapana.
hapana. Alright. Ndanjiti ndiwe ndiwisise because unokuzowa kuti Juliet rasvika.
Okay, what was your question?
Ndanga ndichibvunza kuti pama-meetings ane zuro nezuro riya and kumashure ndaisange ndiripo.
Did you listen to what I said?
What?
Pandanchiita summary of what happened last time.
Pamatanga nhasi?
Yes.
Yes. Ndandavapo.
So you have not lost anything? Plus, there are also some videos which you can play.
Okay. Ndanga ndichibvunza kuti hapana zvamakapa vamwe like assignment and the likes.
No, not yet.
All right, thank you.
Because how can I give you an assignment before you have even started?
True.
But from now I can give assignment because we have covered at least we have covered the transpiration.
Okay.
So I can give you some assignment on the transpiration just to check whether you are understanding some concepts.
Okay.
Yes.
Thank you.
Hello, Doc.
Okay, thank you very much.
No, Doc, I have a question, Doc.
But how large is this class? How many are you? Do you know?
Not
Around 200.
Do we have a class rep?
Kuzi 200 ogona kusvika because vamwe havasi kuita ka-LS.
Say, I will confirm and get back to you with the accurate number.
Okay, because normally we don't exceed the 50 when you are doing these lectures. And if we are 200, we could be losing some students, you know. Anyway, let us meet tomorrow at 2 o'clock.
Na Dr. Kumbiroga, this recording togona kuiwana sei.
Near nezuro foot.
Yeah, I'll put some of these recordings on your
Class email.
Yeah, on the no matter I put them on YouTube.
On YouTube, okay, that's okay. Then.
Yes. So I'm going to give you the links.
Yeah, okay, thank you, Doc.
Right now.
Class email say.
Yeah.